Hi girlies! So I've been doing a lot of research on this thing called DNA methylation, and I read a lot of articles on it, a lot of research articles on it, and I was like, wouldn't my life be so much easier if someone just girl bossed this and explained it to me? So that's what I'm gonna be doing. I'm gonna be girl bossing DNA methylation and dumbing it down so that someone like me doesn't have to read 16 pages worth of research articles. Let's get into it. So basically, let's break this down. There's DNA and there's methylation, right? So DNA, I'm sure we know, is like the genetic material information of our body. It like tells our body what to do. It tells our body how to act. It's like a group of popular girls at our school. And DNA methylation is like broken down even further into these little things called nucleotides. And like nucleotides are made of like phosphate groups, 5-carbon sugars, and nitrogenous bases. So like our nitrogenous bases are like further broken up into two groups. So there's 4 nitrogenous bases and they're broken up into 2 groups. I can't do that fancy V thing. So there's purines and there's pyrimidines. Picture of it like boys and girls almost. So the purines are adenine and guanine, and the pyrimidines are cytosine and thymine. And some of these nitrogenous bases get along with others, while some of them don't. So the more they get along, the stronger the bond between them. It's like a relationship in a way. And so how the bonds work, the best possible pairings, like OTP meant to be, are guanine and cytosine and adenine and thymine. So for our sake, we're only going to focus on cytosine and adenine because the rest of them are kind of not relevant. But yeah, that's like the DNA side of things. Let's talk methylation. So like methylation is basically adding a methyl group to any organic compound, any molecule, all that fun stuff. And I'm sure you're like, what is a methyl group? A methyl group is basically a molecule that's made up of 1 carbon and up to 4 hydrogens. So carbon, hydrogen 1, hydrogen 2, hydrogen 3, hydrogen 4. That's max. What DNA methylation does is like, remember I told you the DNA is like in charge of everything? DNA methylation is like Regina George. Basically, it'll come onto you and it'll be like, shut up. Like, don't even speak to me. And that entire gene just silent. It's like Queen Bee up to, like on top of the world. So what it does is the methyl group gets added to the DNA, and depending on where it is in the DNA, it can silence the entire gene. So let's say like you have a gene that gives you like a fifth finger, a 6th finger. If the methyl group goes and attaches itself to the first part of the gene that gives you that 6th finger, the entire gene is going to be silenced and you'll only have 5 fingers. So yay you have 5 fingers. DNA methylation can increase gene expression, but we've noticed for us in like cells that do not create the cells of our children, DNA methylation normally silences genes instead. So as I mentioned earlier, DNA methylation occurs mainly in cytosine and adenine, but like where in our body does it occur? So we've noticed that like when we're like babies, like embryos, still in our mother's womb, there's a lot of DNA methylation there. But as we grow older, our tissues mature and like cell differentiation happens, like cells get different jobs. DNA methylation kind of decreases and everywhere except for the brain. So our brain has a lot of DNA methylation occurring up there. Why? We don't know, because the brain is kind of complicated and we don't really understand anything it does. So we know that it exists, but like why? That's a good question. That's a really good question. Other than that, one place DNA methylation is like super prominent is on something called CPG islands. So I'm gonna get into this a little bit later, but cytosine is the most methylated nitrogenous base that we know of. So a lot of our research is dedicated to that. And we noticed that on like pairings of nucleotides that have guanine and nucleotides that have cytosine, that true pair, that OTP, they are more likely to be methylated compared to like any other pairing. So another reason why they're really good for each other. we noticed DNA methylation in two nitrogenous bases, as I mentioned earlier, adenine and cytosine. So we just recently found out that adenine can be methylated in humans and mammals. So we've only just started researching it, but we do know that there's only currently one form of methylation for adenine, and that is to create N6-methyl adenine, or CMA for short. In cytosine, we know of two ways because of how much we've researched it, and that is N4 methyl cytosine, or as 5-methyl cytosine. And because 5-methyl cytosine, or 5MC for short, accounts for 1% of our nucleic genome, we've researched that a lot more. So like literally everything I'm going to be saying is related to that. So now that we've kind of understood what DNA methylation is, where it is, and all that, how do we actually make that happen? So attaching the methyl group to the cytosine in our scenario is actually a really complicated process. So we have these enzymes which like speed things up, and they're called DNA methyltransferases, and that's a really long word to say, so we're just going to keep it short and simple and call them DMTSs instead. So they're really helpful. There's like 4 of them. There's DNMT1, DNMT3A, DNMT3B, and DNMT3L. So let's break them down. DNMT1 is kind of like the maintenance DMT. So basically, when cell replication occurs and like a parent cell gives birth to a daughter cell, typically they're supposed to be identical, except that DNA methylation doesn't really get replicated super well, so it kind of just disappears. But DMT sees it and it's like, yo, dude, like my brothers just worked on that. I'm not gonna let you mess this up for them. Like they worked hard on this. So DMT goes in, copies the DNA methylation in the parent cell and pastes it onto one strand of the DNA of the parent daughter cell. And then, you know, the daughter cell grows up, she becomes a nice adult, and DNMT1 looks at her and she's like, yo, why are you only methylated on one strand? Like, dude, I just told you my brothers worked hard for that. What? Like, what is this? So it's like, I gotta do everything in my house. And it goes and it fixes that daughter cell so that it's not hemimethylated. It has DNA on both strands instead of just one. DNMT3A and DNMT3B are also called de novo DNMTs, which is like Portuguese for a fresh new tip for you guys. It's a pretty good name, mainly because you know how it's like DNMT 1 is like yelling at everyone that its brothers are working hard and no one cares. Well, DNMT3A and DNMT3B are the ones who really, you know, make the magic happen. So like unfortunately there's no seagull that's gonna come and drop DNA methylation to the DNA's front porch. You know, someone's gotta do the work here. So DNMT3A and DNMT3B go in and they add methylation because they're like, this gene, like I don't really like her vibe, like she's just not giving off with her head. That's basically how it works. DNMT3L is kind of like the black sheep of the family, mainly because it can't bind to DNA, so it can't actually be an enzyme. It's like, it just can't. But what it can do is it can encourage DNMT3A and DNMT3B to be better at what they do and actually make sure that they're getting the job done. So it's like a supervisor. It's a manager at the company of DMT. So it basically just makes sure like they're on track, they're doing what they need to do, they're not like getting any upset customers, they're not burning the company down, basically it. So now that we've kind of understood how to make DNA methylation, let's talk how to demake DNA methylation. How do we demethylate it? So there's 2 ways our body can do that, either passively or actively. So remember how I mentioned, let's talk about passively first because it's like so much easier to understand. So remember I said earlier DMT1 is like the maintenance DMT, and it goes in and makes sure that like the DNA methylation generation continues. So sometimes there's like not enough DMT1, or it's like busy being inhibited by other things. It's just like busy. It's taking its like lunch break or something. It's just not in the mood to work. It's on strike, you know, so that when that happens, DNA methylation doesn't continue from one generation to the next, and the family line just ends there. So the body accidentally gets rid of DNA methylation that way because there isn't enough DNMT1 to continue the process. So actively demethylating DNA is pretty complicated, mainly because it's not a good sign. So when our body, for example, one instance that we notice that DNA demethylation is decreasing, DNA demethylation is increasing rapidly, is when there's a cancer in the body. So DNA demethylation, active DNA demethylation, is normally a red flag. And it's also a really complicated process because there's no direct way to turn 5-MC in particular back down to cytosine. But like, don't worry, I got you. We're gonna go through all the different ways. So the first way is by taking a cytosine and adding something called AID/APOBOC, which I'm not gonna give the full form for because if I do, we're gonna be here for a whole extra minute. And I also can't say the full form. So but basically what that does is it changes the cytosine to a thymine or a uracil, in our case a thymine, and the body's like, thymine and guanine, like that's not like, they don't have a strong bond, they're not giving, it's like a really bad relationship. So basically the body plays the act of a desi parent and being like, you cannot marry her, and it takes something called a TDG, which basically changes thymines into cytosines, and boom, problem solved. The second way to demethylate 5-MC is by using a lot of different methods. So the first one is first step is using something called TET. And what TET does, TET, is that it adds a hydroxy to the cytosine and it makes it 5-HMC instead. And then the body's like, hey, APOBOC, can you help us out here? And APOBOC, AID, go in and make this 5-HMC into 5-HMU, so into a uracil. And the body again is like, dude, like, what is going on? Like, you cannot, like, this is an improper pairing. You guys cannot marry each other. Like, where are her parents? They call TDG. And a new friend of ours called Smug1. And Smug1 is very smug about its ability to change 5-HMU back down to cytosine. So that's way number 2 to demethylate cytosine, is 5-MC. The third way to demethylate 5-MC is similar to the second. So 5-MC uses TET to become 5-HMC, then uses another TET to become 5-FC. And then from here, there's like 2 options. So option number 1 is by using a TDG to go back to cytosine, or by using another TET, which is option number 2, becoming 5-CAC, and then using TDG to go back to cytosine. Why it can possibly has the option to use another TET, I don't know. I don't like it. But yeah, so that's how we demethylate 5-MC. And that's really it. I hope you guys enjoyed. I hope this was easy to understand and much easier than going through a bunch of articles. So I'll be honest here, I did summarize a lot of the stuff in this little section, this little video, but I do have a longer and more detailed explanation in an article that I wrote. I would highly encourage you guys to check it out. I'll put it in the link below, the description box below. I feel like a YouTuber. That being said, feel free to like if this video helped you out. Smash that subscribe button. Sorry, I had to do that once. Smash that subscribe button. And yeah, I'll see you guys next time. Bye.